My name is Gabriel de la Fuente. I'm coming from the University of Lleida. I'm going to try to explain a little bit this study that is belonging to the FIDAGIN project, specifically in the Work Package 2. The aim of this Work Package is try to identify different biomarkers or indicators that will be linked to the performance of the animals. In this study, what we did was try to see whether the microbial composition of the fecal samples in our study in pigs was in somehow related with the performance. So for doing that, we use two different productive types, the pietrain and duroc, and two different diets. One with a normal level of protein, around 13% crude protein and finishing, and another one with a higher level of protein, the 15%. Then we perform all this analysis to see which was the digestibility of the nutrients, feed intake, average daily gain, feed conversion rate, and so on. And then also from the fecal samples, we extracted the DNA, and then we analyze it through next generation sequencing to see the microbial characterization. The results from the performance analysis, they are not shown in this poster, but briefly, there was no differences in the feed intake between both production types, but there was a difference in the average daily gain and the feed conversion rate. These two different productive types, they had a different efficiency in the utilization of the food. Then we tried to see whether there was differences also in the microbial composition. So part of the results are shown here, and we indeed found differences in the main groups, in the Firmicutes, Bacteroidetes, and also this ratio that is quite important in nutrition, the Firmicutes Bacteroidetes ratio in between two breeds, but we didn't see any differences between diets. And then going a bit deeper, we saw some differences in families like the Lactobacillus, Clostridium, and also some differences due to the diet that is normal, because the diet in somehow is going to influence the microbial population. The last step of this study was try to join both sides, the performance and the microbiota of these animals, and try to see there was kind of a relation or potential biomarkers. For doing that, we performed a canonical correspondence analysis, in which we can see in this graph that these dots correspond to animals, but this microbial population in a multivariate analysis of those animals, and the arrows, they correspond to the environmental variables that in our case were the performance. We only saw the significant ones and the ones that they clearly were related with the microbial population. And then these circles are the confidence intervals of these four groups that we studied, both duroc and pietrain in high and normal protein diets. What we can see in this graph is that the emission of ammonia was clearly related with the breed, being the duroc, the breed or the productive type that was producing a higher amount. Some of the traits that we found to be linked to the microbial composition was the starch digestibility, crude protein digestibility, and also some CO2. So in somehow we could demonstrate that indeed there's a clear link between microbiota and performance. Now the next step is to identify individuals, so we can just in somehow early detect and then classify animals for high, low efficiency, just using the microbial population as bioindicators.